Associate Professor in Department of Biotechnology, Centers of College of Engineering. So today we are going to discuss about a genetic engineering experiment which is called blotting. So before checking out about blotting, we will see what is meant by blotting technique. So the blotting term refers to transfer. So it's nothing but we are going to transfer a band, either DNA or a protein, from gel towards MCM membrane. So here the MCM membrane refers to nitrocellulose membrane. So this transfer refers to, that is from gel towards your membrane, refers to the term blotting. So this blotting can be classified into three types. One is your western blotting, other one is your southern blotting, other one is your northern blotting. So in western, a protein will be detected. In southern, a DNA will be detected. And in case of northern, an RNA will be detected. So if you want to detect a protein sample, this protein should be first run towards your SDS page. So this is sodium dodecyl sulfate polyacrylamide gel electrophoresis. This helps in separation of protein. So you will be loading n number of proteins. From this we are going to screen only a single protein. So for that we use as a confirmatory western blot. That is, this protein can be detected by means of an immunoscreening. So immunoscreening here refers to the interaction of an antigen with antibody. So here the antigen is your protein to be detected, and antibody is created against this protein. So this detection helps in screening of a single protein from n number of your proteins. So next comes your southern blotting. So in this southern blotting, I am going to check my DNA presence. So I am going to run my agarose gel electrophoresis. In this, the DNA will be separated, and this bands, which is numerous, we are going to track again only a single band. So this helps in particular gene isolation, which refers towards your confirmation of presence of a single genomic DNA. Similarly, RNA can be separated using again agarose gel electrophoresis. So again an n number of RNA will be present. From that, a single RNA will be separated. So these blotting techniques plays as a confirmatory test for identification of a single protein or a single DNA or a single RNA sample. So this blotting plays an important role in genetic engineering experiments. So once when you clone, this cloned gene can be confirmed with the help of blotting. So this blotting plays a vital role for final confirmation of a protein, DNA and RNA. So let us see this as a proof by conducting an experiment related to Western blotting and Southern blotting in our laboratory. We will continue the experimental section. About your experimental setup related to blotting. So we have a power pack here. The main goal of power pack is we will get an alternate current, AC current. So when you use for gel electrophoresis, this AC current won't give a refined band. So what we have to do is we have to convert as a DC current. For that we have a rectifier in this power pack. So this rectifier will convert the AC current to DC current. So this will have a setup of volt adjustment. So you can have it as a power where the volt can be set up. So varying volt can be adjusted. If it is an agarose gel electrophoresis, we will go from 50 towards 100, and if it is an SDS page, we will go from 100 to 150. So this setup will be used for providing the DC supply. And you have here the cathode and anode. So you can have the connectivity from the power pack towards your electrophoresis tank. But before running this electrophoresis tank, you have to prepare. If it is an Western blotting, you have to run an gel electrophoresis. So this is vertical pipe. You have the gel tank where you can fill your buffer. You can place your glass in between where the gel has been loaded. So that gel can be kept in between, and then you have to screw it. After screwing it, you have to pour the buffer. So once you pour the buffer, you can load the sample with the help of the micropipette. Whatever volume that is being required, that you have to keep on loading. So once you load it, you set the current what is required in the power pack, and then you can go for running for minimum 1 to 2 hours. So once you complete this process, you will get a gel. So that gel you have to remove it. After removing it, you have to get set up in a blotting. So here we have the anode as well as your setting. You have a scrub. So over that you can keep a sponge, and after this you have to keep your filter paper. This filter paper can be soaked in the buffer, folded in required size. You can go for placing it. So once you place it, you have to place your gel. After placing the gel, you have to again go for your sponge, and then your scrub, and then again your electrodes. So what you have to do is you have to screw it in opposite direction parallelly. So you will get a setup of your connectivity in which the Western blotting can be run. So you have to put this in the tank. So once you put the tank, you can pour the buffer. So this you allow 150 degree or 120 degree centigrade. You can run it. So once you pour the buffer and run it, you can remove. Again while you remove, you have to remove one by one. So once you remove it, you will get from the gel towards your membrane the transferred band. That transferred band should be checked for your primary antibody addition, secondary antibody addition, and then confirm with the bands that has been present. So similar way, we can go for our Southern blotting. Here we have a tank in which weight should be placed. So same setup, you can go for your Southern blotting. Same, you can keep your sponge, filter paper, etc. Then you place your agarose gel, then your nitrocellulose membrane. Again you keep your sponge, scrub, and then you should keep weight minimum 500 gram and maximum 1 to 1.5 kg. So once you keep this setup for whole night, from the agarose gel, the bands will get transferred towards your membrane. So next day, after whole night, next day, by means of capillary action, you will be having the transfer of bands from the gel towards your membrane. So you have to remove it and then take the band. So this band can be identified by means of adding your corresponding DNA or RNA as a probe. Once this has been done, your DNA confirmation will be over. So a similar setup can be done for your RNA experimentation. Through this experimentation, we can confirm that the blotting can be implemented for protein, DNA and RNA. Thank you. Today we will discuss about the genetic engineering experiment PCR. So PCR here stands for polymerase. So P for your polymerase, C for your chain reaction. So your polymerase enzyme is going to produce a DNA chain. So this total reaction we are going to prove using an experimental model, that is called your PCR. So in PCR, three different things will be focused. One is your denaturation, second one is your annealing, and third one is your extension. So we can call this as mechanism. So the PCR works in three steps. We can call this as step 1, this as step 2 and this as step 3. So here in denaturation, if you have a double-stranded DNA, your primer cannot sit on this DNA, so we have to open it to a temperature above 95 degrees centigrade or between 90 to 95 degrees centigrade. This DNA will get denatured, so you will get a single-stranded DNA. So this single-stranded DNA will be allowed to interact with a primer. So in general we have variety of primers. Here I am going to tell you about a forward primer and a reverse primer. So always the direction of synthesis will be from 5' end to 3' end. Again here from 5' end to 3' end. So you have the hanging bases in forward primer as well as you have the hanging bases in reverse primer. So again this is going to interact here. So in case of your annealing, around 55 to 65 degree centigrade will be used for annealing. So the primer will go and attach over here. So this is your primers. So where we have the attachment. So suppose if this is your 3', this will be your 5', this will be your 3', and this will be your 4'. Similarly, this will be your 3', this will be your 5', and here it is 5' and 3'. So both your forward primer and reverse primer will get attached towards your DNA and get annealed. So once it is getting annealed, this strand should be synthesized. So you will be having the extended DNA. So here we have the complementary which is being synthesized using your extension. So now a single strand, that is a duplex form of strand, is going to get converted into two different DNA. So which is, if it is 5', this will be your 3', again 5', 3'. So this is one set of DNA. Again the other set we have similarly, if it is 5', this will be your 3'. If it is is 5 prime, this will be your 3. So you are going to have from a single molecule, 2 molecules of DNA will be generated. So when you set your PCR, the same set of steps will be repeated. So once when you repeat n number of cycles, or roughly 30 to 40 cycles in a lab, you will get numerous copies of DNA. So this numerous copies of DNA can be identified by running agarose gel electrophoresis. So this agarose gel will prove whether the DNA has been amplified or not. So you will be having an agarose gel. So when you load, you have to load in the wells. And the first well you will be preferably loading your marker. And the remaining wells you can go for your positive. It may be sample 1, 2, 3. And you can go for your negative sample loading. So to check whether the reactions are free from impurities. So in marker, suppose if you are going to load a 100 base pair marker, you will be getting around 10 bands. Suppose if it is 100 base pair. Okay. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So here you have your 1000 base pair, or you can call it as 1 kb. So this, when you load with your sample, in negative you won't get any bands, but in positive, suppose if my band size is 800 base pair, I will be getting a positive band of 800 base pair. So this proves that my DNA sample has been identified, this has been denatured, this has been annealed, this has been extended. So this extension temperature will be 72 degrees centigrade. So this will be repeated for 30 to 40 cycles. So I will be getting n number of copies. That n number of copies can be proved with agarose gel electrophoresis. And this agarose gel electrophoresis is a proof for PCR confirmation. Let us see the same your PCR. So we have here a PCR machine. So this PCR machine is used for amplification of the DNA. Suppose if I am going to isolate a genomic DNA from leaves, okay, I will be making this leaves into pieces, and this leaves will be grinded. So once it is grinded, it will be put in suitable solvent. From this, the DNA sample will be extracted. This extracted DNA can be used as a source for your DNA amplification, or you can use any cloned DNA, that is your gene of interest, can be used as a template to get amplified. So when you go for your isolated DNA in a purified form, this can be amplified into numerous copies. So when you see this PCR machine, so this is an biorad PCR machine. So it is otherwise called a thermocycler. Why it is called a thermocycler? We are using different temperature. So we have here a thermal maintaining block. Okay. So this gives you the 90 degree, or your 50 to 65 degree, or 72 degree. This thermo blocker will help us to maintain. And here you can see the tubes. For example, when you have the sample DNA, this sample DNA can be put into the machine. Once you fill it with your template, your required buffer, your polymerase, as well as your magnesium, that is your salt, you have to close it and then keep it in the PCR machine and go for the setup. As we have said, there is 3 main steps. One is your denaturation, where it occurs in 90 degrees, and next one is your annealing, so between 55 to 65, and next one is your extension. And as I said, we have to repeat this for 30 to 45 cycles. You have to go to step 2, that is you have to go for repetition. Okay, this is your initial denaturation and this is your final extension, and after this you have the holding. So this holding can be 4 degrees centigrade even. So you have to go for the seconds also setting. Once the required temperature and required settings has been done, you have to place the tube and then close it, and then you can go for the starting of the experiment. So this will simplify how the single DNA can be multiplied using this PCR machine. So this PCR machine plays an important role. It has variety of application, especially when you have the forensic sciences sample. You will be having only in small quantity. So those samples can be amplified. In case of corona, we need for RT-PCR, where we have the coronavirus. The RNA is being converted into cDNA, and this cDNA will be amplified. So once the amplified DNA has been done, you can find out whether the patient is having positive or negative. So the main goal of this is, when you have the sample as here, lesser quantity, this quantity can be improved with the help of the PCR amplification. So you need the template properly. In purified form, it will be better, you will have a good amplification. Along with that, you add your enzyme polymerase, which is going to extend it, and you need a primer which is going to initiate the reaction, and you need a buffer which keeps the DNA as well as your enzymes in an active form. And finally, when you repeat this as a denaturation and your annealing and extension, your amplified product can be applied. And this amplified product, once the reaction is over, you have to take this. You should not touch here because it will be in hot condition. So you have to take this tube, then you have to go for agarose gel loading. Once you load it, you will get a confirmation. That is, if the bands are present, you can say it is positive, you have got the PCR product. If the bands are absent, you can go for repetition. That may be due to failure, that is annealing failure. You may have false, or you may have primer dimer, again you may have the false result. If there is template, your DNA source of primers as well as your polymerase activity is good, you will get a clear product. So this can be obtained as a band in which compared with that of the marker DNA. Thank you.
scope of the present work is we want to produce a metal and chemical free CQD, carbon quantum dots, in order to degrade the organic dyes, reactive dyes, and the traces of dyes which is present in the textile industry effluents. So our applicability is for the industry purposes, the treated water to be reused. So the hypothesis of our study is we want to generate a photocatalyst of green chemistry approach, which is devoid of chemicals as well as it is going to produce a treated water which is of dye degradation, free of chemicals. So the hypothesis what we want to highlight is the CQDs after getting exposed to the sunlight, it goes to the excited state. This excited state is capable of producing the activated hydroxyl ion, which is capable of breaking down the pollutants into smaller molecules, thereby the dye degradation results. So this can be proved through our studies using malachite dyes. So the complex molecules have been broken down into the simpler molecules. Then we went the same sort of studies with the help of reactive rate, M8. So this also we have done through GCMS analysis. Initially, we had the complex set of molecules, and finally, we have got the degraded simpler form of the molecules. So the roadmap of the study is we have taken the microalgae, prepared CQD using the micro oven technique. So after that, this micro assisted technique has been given the product. That product is being characterized. So the characterization has been done. After that, it has been focused for the different applications. So after the applications has been satisfied, we have done one futuristic application for bioimaging too. So this is the overall methodology what we have adapted. So we have taken the spirulina powder, we have prepared the slurry, we have converted into CQDs, and we have characterized. After characterization, we have went for various dyes degradation. So this is the outline of our malachite green. So we have got almost 90.5% degraded water. And in case of reactive rate, we have got 95.6% of total degradation. And in case of mixture of dyes, reactive dyes, 95.2% we have achieved. So in case of the effluents, we have got three different companies. Effluent A, 94.2%, effluent B, 97.3%, and effluent C, 91% of degradation. So a high percentage of degradation was achieved. So the characterization studies related to CQDs have been done. In case of UV visible spectra, we have got a peak of 240 to 295. It gives the presence of carbon, oxygen, and carbon-carbon. And in case of PL spectra, we have proved a maximum lambda max at 480 nanometer, a sign fluorescence. And in case of FTAR, the alkene and alkyne band's presence has been proven. So through XRD and XPS, we have proved the presence of the size of the molecule as well as the carbon-carbon bonds. And in case of TEM and AFM analysis and particle size analysis, we have proved the size is between in the range of 10 to 30 nanometer. And the biocompatibility study, what we have done is using CQD sample, malachite dye, as well as degraded water. So we can see in case of CQDs and degraded water, 100% it correlates with the control. But in case of the dye which is directly treated, we can see the death rate. That is the compatibility is being less with the malachite green. So in case of hemolytic assay, we have got very good results. There is no hemolysis for the CQDs what we have used in the sample. It almost cooperate with the control sample. And we want to prove for the usage, reusage of water agriculture purposes. So there is no interaction of CQD with the mitotic division. So we have proved this study for reusage in the agriculture purpose. So once if the treated water is discharged for the growth of fish, so whether it is compatible or not, we want to prove it. So this study has been proved that the CQDs are non-toxic to the zebrafish. So this is the reusability study. What we have checked is the green gram as well as the spirulina cultivation. And we have collected the samples from various industries which is present in Tirupur, Rasipuram, Kanchipuram, etc. So these are the total quality analysis of the treated water. So it goes with the standards which has been given by the Central Pollution Authority. And this is the lab what we have done through the DST fund recurring. And this is the innovation what we have achieved. That is from the microalgae we have prepared the liquid form of CQDs and powder form of the CQDs, and both the forms has been applied for the treatment of the dye degradation. And the linkages what we have developed through this is technology transfer, product testing for the technology transfer. We have filed 2 patents, and the medical services what we are focusing is the bioimaging study. So the picture shows the control samples of the fibroblast, and here the CPD is treated. So you can observe the dye which is giving fluorescence under the fluorescence method. So these are the 2 patents what we have published techniques. So this is our overall conclusion that the prepared CQD can be used for any type of dye degradation, especially this is discharged through the textile industries. So it is going to helpful for the industrialists to release the zero discharge of dyes through the effluents, and it can be highly benefited by the rural and urban peoples. And this product can be in future can be used for the biomedical, smart solar panel, and optoelectronic applications. So these are the references we have used, and thank you very much for listening.